For those of you who don't know me, I started teaching. I'm the program director for the first and only existing Integrative Cancer Fellowship in the country. And the purpose of this lecture is just to expose everybody out in the audience to integrative cancer therapy. For advanced stage cancer, there is not one cure. There's not one modality for treatment. And so we're going to address an integrative approach to cancer. Okay, well, let me start with the definition as listed by the American Cancer Society, the definition of cancer. According to the American Cancer Society, cancer is a group of diseases characterized by uncontrolled growth and spread of abnormal cells. I disagree with that definition. In the past 40 years, we have minimally extended survival for most solid tumors. But it's not uncommon for new drugs to reveal spectacular results by curing cancer in mice. And that's great because certainly cancer is an epidemic in mice, and that's great that we're taking care of the mice. Unfortunately, it's generally not translating to humans. So you have to ask this question. If we are curing cancer in mice, are our models through which we research cancer in the laboratory valid? So how do we do mouse studies or animal studies? Well, for a long time, we've been doing the human tumor xenografts. And that's where you'll take cell lines that have been growing in a lab and for years and years, and they've mutated. And then we'll typically inject them subcutaneously in the animal. And then what we'll do, and these animals may be immunodeficient, so it'll support the growth of a tumor. Very often, this is not cancer. And what I mean by that is it's a local growth that very often doesn't even metastasize. And then we may treat it with a chemotherapy drug, and we see that local growth shrink. And we say, okay, wow, this is a successful drug. But then you take it to the human, and it doesn't work. And it most likely doesn't work because the model is not accurate. Well, we also use transgenic mice or transgenic animals, and that's where we will insert a gene from another animal, maybe the same species or a different species, and that gene may confer a predisposition for developing a certain type of cancer. So we'll inject it into the fertilized embryo. And this way the animal spontaneously develops cancer instead of us injecting cancer. So we thought maybe that's a better model, and we're still doing that. But unfortunately, when we see good results in the animal, that usually does not confer much with regards to a good result in humans, because we're looking generally at a single gene or gene mutation. And that's not what cancer is. Cancer is not just one single mutation. And another technique called orthotopic transplants, that is probably a better model. That's where we actually excise the cancer and surrounding tissue from the patient and directly transplant it to the animal. And that way, we think it's a better model because we have some of the environment. It's very painstaking, it's more costly, and it's not done that often. So in general, what I'm going to say, and I think that most cancer scientists would agree, is that our animal models are not wonderful. And that's why even when we cure cancer in the animals, it usually does not mean a cure in patients. Okay. Now, again, the definition, according to the American Cancer Society, cancer is a group of diseases characterized by uncontrolled growth and spread of abnormal cells. Well, here is the definition that I propose. Cancer is the interaction between abnormal cells and its environment, and its natural environment, that results in uncontrolled cellular growth. So what's going on is that cancer cells are constantly communicating with the environment. And in the next slides, we'll talk about what the environment is. So if you just take cancer cells and you transplant them into another animal, but you're not moving the environment, it's not cancer. Cancer is the constant communication between these abnormal mutated cells and its natural environment. So we have spent most of our time and money attacking cancer cells while ignoring the environment, which communicates and governs the actions of these abnormal cells. So what is the environment? The cancer environment is the bone marrow. The bone marrow consists of the innate and the acquired immune system. The innate immune system consists of neutrophils, macrophages, dendritic cells, and natural killer cells. And the acquired immune system, the T lymphocytes, B lymphocytes, and antibodies. Well, we were taught a long time ago that the acquired immune system is really what's most important in taking care of cancer. But in fact, now we have much more evidence to say that the most important is the innate immune system, the neutrophils and the macrophages being very important, as well as the antigen-presenting cells and natural killer cells. Okay. Well, also what's important are the endothelial progenitor cells. Endothelial progenitor cells are literally recruited and signaled by cancer cells to migrate to another region so they can then undergo or assist in neovascularization or angiogenesis. So there's signaling going on, and the platelets are involved in that as well. Well, also in the environment is the extracellular environment. As you probably are aware, a lot of cancer relies on glycolysis, and they produce an acidic medium because they pump acid outside the cell, and the surrounding tissue is acidic. Now, the acidic medium outside the cancer promotes angiogenesis and angiogenic factors. And then there's the energy supply. We talked about many tumors, probably 90 to 95% of cancers, are glycolytic, meaning they rely on glucose and glycolysis. The rest of the cancers may rely more on fatty acid metabolism, synthesis, and metabolism. So that's another way to affect cancer, by affecting its energy supply. So we've spent so much time coming up with another cytotoxic drug to kill another cell, another pathway, another receptor, but ignoring the big picture. Now, some of you may have attended the conference where Dr. Shui and Dr. Maharaj were lecturing. And certainly Dr. Maharaj talks about stem cell transplants. They're both involved right now, a very small study because of lack of funding, leukocyte infusion therapy. Now, here is a technique where we're harnessing the immune system to do the job. Now, this makes a lot more sense than just targeting a signal pathway. And just to summarize quickly, for those of you who are not familiar with Dr. Shui's work, is he serendipitously found that some mice are innately immune to cancer. He tried giving a mouse cancer, and no matter how hard he tried, he couldn't get, that mouse would not develop any growth. And so what he found is that mouse was 100% resistant to cancer. And he breeded that mouse with other mice, and approximately 40% of the progeny were also resistant to cancer. And what he demonstrated was that it was, in fact, the neutrophils and the macrophages that encircled the cancer cells and lysed their membranes, and they were responsible for killing cancer. So he ended up developing an assay